It's Chapter 30, Conversion of Amino Acids to Specialized Products. And I have to be honest, before I really sat down with this, if you'd asked me what an amino acid was, I would have given the standard answer.
Which is?
You know, the building blocks of protein, muscle stuff.
And that's not wrong.
Uh-huh.
Not technically.
Yeah.
But it's so incomplete. It's a bit like saying a smartphone is just, a flashlight.
Yeah, it has a flashlight function, but that's not really the point of it.
Precisely. It could do 1,000 other things that are probably way more critical to your day-to-day life.
And that is exactly the hook for today's deep dive. We're getting past this idea of amino acids as just bricks in a wall.
Mm-hmm.
They're actually the raw materials for the body's most critical, most active chemicals. We're talking neurotransmitters that let you think and feel.
Hormones that drive your stress response, your sleep cycle.
Pigments that color your skin, even the specific energy molecules that let your muscles fire instantly.
It really is a form of alchemy. The body takes these basic dietary components, things you get from, say, a piece of fish or a handful of nuts, and it runs them through these incredibly specific enzymatic pathways to create what Chapter 30 calls specialized products.
And that's our mission today. The source text is dense. It is packed. There are 13 different figures, 30:1 through 30:13, and each one is a complex chemical map.
They can be a bit intimidating at first glance.
For sure. But our goal is to try and demystify every single line, every pathway. We want to understand really specifically how something like adrenaline or histamine is actually made.
And just as important, how it's broken down.
Mm-hmm.
Because in biology, the off switch is just as critical as the on switch.
Right. If you can't turn these signals off, you get disease.
You get chaos, yeah.
So to keep us from getting totally lost in the chemical soup, we've broken this down into functional groups based on what these amino acids produce.
Good roadmap.
Yeah, we'll talk about the master regulators, the molecules for growth, the energy and detox crew, the mood and sleep factory. We've got a lot to cover.
We do. And we should probably start with one of the most versatile players on the entire field.
That would be arginine.
Arginine. It's a non-essential amino acid, which means, our bodies can synthesize it.
But non-essential is a bit of a misnomer, isn't it?
It is. Under certain conditions like stress or illness, it becomes what we call conditionally essential. Our internal production just can't keep up with demand.
So we need to get it from our diet. Okay, so let's look at figure 30:1 from the text. This is the big overview, the metabolic fates of arginine. And wow, this guy is a multitasker.
It really is. It's like a central hub at a busy train station.
That's a great way to put it. You've got arginine right in the middle, and then you see these tracks shooting off in 4 totally different directions.
4 major, distinct, and crucial systems. The first one listed is urea synthesis.
Which is basically waste removal, getting rid of toxic ammonia.
Correct. We've touched on the urea cycle before. It's how we package up excess nitrogen to be excreted safely. But look at the other arrows.
Okay, I see one going to creatine synthesis.
Pure energy storage for muscle.
Another one to polyamines.
Which are absolutely essential for cell growth and division.
And then the fourth one, which has gotten a lot of attention in recent years, nitric oxide.
This signaling molecule.
Let's unpack that one first, because figure 32 is dedicated entirely to its synthesis. Now, I think when people hear nitric oxide, their mind might go to the dentist, laughing gas.
A very common confusion. That's nitrous oxide, N2O. This is nitric oxide, just NO.
Totally different molecule, totally different function.
Worlds apart. Nitric oxide, or NO, is a gas. And it's a signaling molecule, one of the most unique ones we have.
So how do we make it? The figure points to an enzyme called nitric oxide synthase. The text is so specific, it even gives the EC number, 1.14.13.39.
I love that level of detail. And it describes the reaction as a 5-electron oxidoreductase reaction.
That sounds intense. What does that actually mean in plain English?
It means it's a very, very powerful chemical reaction. Think of it like a controlled explosion. You're performing a really difficult piece of chemical surgery.
Okay.
You start with arginine. It has this part called a guanidine group. It's a specific arrangement of nitrogen and carbon atoms at the end of the molecule.
Right.
The enzyme, nitric oxide synthase, has to basically reach in, grab one of the nitrogen atoms from that group, and rip it off.
Just pulls it right off the molecule.
Pulls it off and combines it with an oxygen atom from the air we breathe. To do that, to tear a nitrogen away from a stable structure and bond it to oxygen without, wrecking the whole cell.
You need a lot of energy and control.
Exactly. You have to move 5 electrons around simultaneously. It's a high-wire act of chemistry.
And it's not a solo act, is it? The text says it requires a whole bunch of cofactors.
A whole team. It consumes NADPH, which is a source of electrons. It needs FAD,
Yeah.
FMN,
Heme,
like
hemoglobin.
the very same, the iron-containing group. And it also needs something called tetrahydrobiopterin. It's a very, very expensive reaction for the body to run.
So what's the end product? What do you get for all that effort?
You get two things. The arginine molecule, now missing a nitrogen, becomes L-ornithine. And the nitrogen that was ripped off becomes the gas, nitric oxide.
And that L-ornithine, it doesn't just get thrown away, does it?
No, nothing gets wasted. L-ornithine can be recycled right back into the urea cycle or used for other things, as we'll see. But the real star of this show is the nitric oxide.
So why go to all that trouble? Why make this gas?
Because it's a ghost. It's a tiny, uncharged gas molecule, which means it can pass straight through cell membranes. It doesn't need a receptor on the outside of a cell to deliver its message.
Just floats right in.
Floats right in and talks to the machinery inside the target cell directly. It's an intercellular signaling molecule, a phantom messenger.
And what's the message it's usually delivering?
In most cases, the message is relax.
Relax.
It's a potent, smooth muscle relaxant. Think about your blood vessels. They're lined with smooth muscle. When no diffuses into those muscle cells, it tells them to relax, and the blood vessel expands.
It's a vasodilator.
A very powerful one. This is crucial for regulating blood pressure. When your vessels dilate, your blood pressure drops and blood flow increases.
So without arginine and this crazy 5-electron reaction, our entire cardiovascular system would be, what, rigid and high pressure?
It wouldn't be able to adapt precisely. It also acts as a neurotransmitter in the brain and plays a role in the immune system. It's regulating blood flow, neural signals, and even fighting off pathogens.
Incredible. Okay, that's fate number 1 for arginine. Now, let's go to fate number 2, which I have to say brings us to some of the strangest names in all of biology.
I think I know where you're going.
I'm looking at figure 30-8 and figure 30-9. We're talking about polyamines, and the names here are, well, they're putrescine, spermine, and spermidine.
Yes. Biochemical nomenclature often gives you a little history lesson. You can probably guess where these molecules were first isolated.
I mean, putrescine sounds an awful lot like putrid.
It's the characteristic smell of decaying flesh, that distinct, unpleasant odor. A large part of that is putrescine being produced by bacteria.
And spermine and spermidine
were first discovered in semen. But despite these, let's say, unsavory origins, these molecules are absolutely vital for life. They're what the text calls biologically active amines.
So let's trace the path. How do we get from our hero, arginine, to something called putrescine?
It's a bit of a relay race. And it starts with L-ornithine.
The same L-ornithine we just made as a byproduct of the nitric oxide reaction?
The very same.
Yeah.
See, the body's already recycling. It's efficient.
So we take that L-ornithine. What's next?
An enzyme called ornithine decarboxylase comes in and performs a very common trick in biochemistry.
Decarboxylase. Okay, so that carboxyl part means it's removing a carbon group, right?
Exactly. It removes a carboxyl group, which is a COH, and releases it as a puff of carbon dioxide, CO2. Imagine just snipping the tail off the ornithine molecule.
Okay.
When you snip that tail off, the molecule that's left over, that's putrescine.
So putrescine is just decarboxylated ornithine.
That's all it is. And now we have our base. We can build up from here to make the others.
How do we do that? What do we add?
We need to add a piece called a propylamino group. It's a little chain of 3 carbons and a nitrogen. We take putrescine and we use an enzyme called spermidine synthase to attach one of these propylamino groups. And now putrescine has become spermidine.
And I'm guessing to get to spermine, you just do it again.
You do it again, you bring in spermine synthase, you slap on another propylamino group, and you get spermine.
So where are these propylamino groups coming from? Does the cell just have a bucket of them lying around?
This is a beautiful connection to another pathway. They come from another amino acid.
Which one?
Meatinine. Specifically from a molecule called decarboxylated S-adenosylmethionine. But let's put a pin in that for a moment. We'll come back to it.
Okay, pinning that. So we've built these molecules, putrescine, spermidine, spermine. What do they actually do? Why does the body spend all this energy making chemicals that smell like death?
Look at their structure. They're long, flexible chains. And dotted along those chains are nitrogen atoms that carry a positive charge at cellular pH.
So they're polycations.
They are polycations. They have multiple positive charges. Now think about the most important molecule in the nucleus of a cell. And what is the overall charge of a DNA molecule?
It's negative, right? Because of all the phosphate groups in the backbone.
Highly, highly negatively charged. And you know what they say about opposites?
They attract.
These positively charged polyamines are like little molecular magnets. They are drawn to the negatively charged DNA and RNA.
And what happens when they bind?
They help to neutralize that negative charge. This allows the DNA, which would otherwise repel itself, to be packed tightly and neatly into the nucleus. They act as stabilizing agents.
So they're like the spools that you wind the DNA thread around.
That's a great analogy. They're essential for stabilizing and compacting our genetic material. Without them, cells can't divide properly. They're critical growth factors.
So the smell of death molecule, putrescine, is actually a fundamental requirement for life and growth.
Biology is full of these little ironies.
Now, figure 30-9 shows the other side of the coin. The breakdown, the catabolism. What happens when we're done with them or have too many?
We basically run the process in reverse. An enzyme called polyamine oxidase gets to work.
And what does it do?
It clips off those propyl amino groups, it breaks ceramine back down to spermidine, and it can break spermidine back down to putrescine.
But the text says there's a cost to this breakdown.
There is. This reaction, this oxidation, produces some potentially nasty byproducts.
Like what?
The main ones are H2O2, hydrogen peroxide, and various aminoaldehydes.
Hydrogen peroxide, the stuff you put on a cut inside a cell. Isn't that really toxic?
It can be, absolutely. It's a reactive oxygen species. It causes oxidative stress. So the very process of turning over these growth molecules creates a toxic byproduct that the cell's antioxidant systems have to clean up immediately.
It just shows you that growth isn't free. It comes with a metabolic price tag.
A very steep one. You can't just have unregulated growth without generating a lot of dangerous waste.
Fascinating. So that's arginine. It helps relax our blood vessels and helps our cells divide. Let's move on to our next functional group. I've called this one the energy and detox crew.
Okay.
We're looking at cysteine and glycine.
Two of the structurally simplest amino acids, but they punch way, way above their weight class in terms of function.
Let's start with cysteine. The text mentions right away that it's a precursor for a part of coenzyme A.
Yes, coenzyme A or just CoA. If you ever studied metabolism, you see CoA everywhere.
The Krebs cycle, fatty acid oxidation.
It's ubiquitous, and you can think of it as a handle. It's a molecular handle that enzymes use to grab onto and move around acetyl groups or fatty acids.
And cysteine is part of that handle.
It forms the most important part, the business end. It's a portion called thioethanolamine, which contains a sulfur atom. That sulfur atom is what actually forms the bond the molecule's CoA carries.
So no cysteine, no functional coenzyme A, no coenzyme A, no energy metabolism.
You'd be in very big trouble, essentially, yes.
But there's another pathway for cysteine that really caught my eye in figure 30-3. It leads to a molecule called taurine.
Ah, yes. Taurine?
I see taurine on the side of every energy drink can at the gas station. It's always marketed with gives you wings or something. What is it actually doing? Is it a stimulant?
It's so interesting how it's marketed because biologically, its primary role is quite different. The synthesis pathway is straightforward.
Okay, walk me through it.
You start with cysteine. An enzyme called cysteine dioxygenase adds 2 oxygen atoms to it, creating a new molecule called cysteine sulfonate.
So, step 1 is oxidation.
Right? Then, just like with ornithine, we have a decarboxylation step. So phenylalanine decarboxylase comes in, snips off the CO2 group, and you get hypotaurine.
Hypotaurine.
And then one final simple oxidation step via hypotaurine dehydrogenase turns that into taurine.
And the result is this sulfur-containing molecule. What does the body need it for? Why is it so important?
The text highlights its role in digestion, specifically in fat digestion.
Okay.
Taurine's main job is to pair up with cholic acid, which is a bile acid that our liver makes from cholesterol.
And bile acids are like the detergent for our gut, right? They break up fats.
Exactly. They emulsify fats. If you eat something greasy, you need bile acids to break that grease down into microscopic droplets that your digestive enzymes can actually work on.
So where does taurine come in?
The text says taurine displaces coenzyme A from the cholic acid to form a conjugate called taurocholic acid.
And what's special about that?
This new molecule, this taurocholic acid, is a much more effective detergent. It's more water-soluble and better at emulsifying fats than the cholic acid alone. So without enough taurine, you would have a really hard time digesting and absorbing dietary fats.
So the marketing on the energy drink is maybe a bit of a stretch.
A bit, yeah.
It's less about energy in the stimulant sense and more about helping you efficiently process the greasy pizza you might have eaten with the energy drink.
That is a very fair assessment. It's digestive efficiency, not necessarily neural stimulation.
Okay, let's switch to its partner in this section, glycine. Figure 30-4 is all about the biosynthesis of hippurate. This feels like a major detoxification story.
It is the ultimate detox story. We are constantly exposed to, and our bodies produce, compounds that are apolar.
Meaning not water-soluble?
Exactly. They're greasy, oily molecules. And that presents a problem for excretion, because our urine is water-based. You can't just dissolve oil in water.
So these things could get stuck in the body.
They could. And the text gives a perfect common example: benzoate, or benzoic acid.
Where do we get that from?
It's a very common food preservative. It's in sodas, pickles, salad dressings, all sorts of things.
So how does glycine help us get rid of it?
The body has a two-step process. First, it has to activate the benzoate, and it does that by tagging it with coenzyme A. That creates benzoyl-CoA.
The handle again.
The handle again. Now the molecule is activated, and the body uses an enzyme to snap a glycine molecule onto it. This creates a brand new molecule called hippurate.
And hippurate is water-soluble.
Very water-soluble. Can easily dissolve in the blood, get filtered by the kidneys, and be excreted in the urine. Problem solved.
That's so elegant. It's like you have this slippery, greasy box you need to throw out, so you attach a big, easy-to-grab handle to it.
That is a perfect analogy. And glycine does this for a huge number of drugs, food additives, and metabolic byproducts. It's a primary conjugation agent for detoxification.
The text also mentions a few other pretty major roles for glycine. It says it provides carbons for heme, purines, and creatine.
Yes, this is incredible. Glycine is not just a conjugation handle, it's also a literal building block for other critical molecules.
What do you mean?
It means the body takes the glycine molecule and physically breaks it apart, using its carbon and nitrogen atoms as raw materials to build the complex ring structures of these other things.
So parts of the heme molecule in my red blood cells are actually made from bits of glycine.
Yes. Yeah. He, which carries oxygen, and the purines, adenine, and guanine, the A and G in your DNA code.
Wow.
Glycine's atoms are literally woven into the fabric of your blood and your genetic code. It sacrifices itself to build the fundamental machinery of the body.
It's the ultimate spare part.
It really is.
Okay, let's move into section 3. This one is really interesting because it connects something like allergies with something like muscle performance. We're talking about histidine.
Histidine, another crucial player.
And first up, the one everyone knows, histamine. We take antihistamines for hay fever or allergic reactions. So how do we make it?
Figure 30-5 shows it's a remarkably simple one-step reaction, very fast, very direct.
What's the step?
You take the amino acid histidine and you use the enzyme histidine decarboxylase.
Here's that word again, decarboxylase. So we're just snipping off another CO2?
That's all it takes. Snip off the carboxyl group and histidine becomes histamine.
But the text makes a point of noting a requirement. It needs pyridoxal 5-phosphate.
Which is the active form of vitamin B6.
So this is another one of these B vitamin-dependent pathways.
Yeah.
If you're deficient in B6, this reaction, it slows down or stops?
It stalls. This pathway and dozens of other critical amino acid conversions grind to a halt without enough B6.
And histamine is what the text calls a biogenic amine.
Right. And it has two really famous functions.
Yeah.
One is its role in allergic and inflammatory reactions. It's what causes the swelling, the redness, the itching.
The misery of allergies.
Exactly. But its other major role is in regulating gastric acid secretion. It signals to the cells in your stomach to start pumping out acid.
Which is why some of the most powerful antacids are called H2 blockers, right? They're blocking a type of histamine receptor in the stomach.
You are connecting the pharmacology directly to the biochemistry. That's perfect. The same molecule that makes your eyes water can also give you heartburn if it's not properly regulated.
Now, figure 30-6 takes us from the pharmacy to the gym. It shows derivatives of histidine that are found in muscle tissue.
Yes, these are fascinating. Specifically, carnosine and anserine.
What are they?
They're dipeptides, which just means they're made of two amino acids linked together. Carnosine is beta-alanine plus histidine.
And anserine.
Anserine is just methylated carnosine. It's carnosine with an extra methyl group stuck on it.
And the text says these are found in high concentrations in anaerobically contracting skeletal muscle.
Right. So think about what's happening when you're doing something really intense, like sprinting or lifting a heavy weight for reps.
You're not getting enough oxygen to the muscle.
Exactly. Your muscles are burning fuel anaerobically, and a byproduct of that is a buildup of acids, specifically lactate, and hydrogen ions, so protons. The pH inside the muscle cell starts to drop.
That's the burn, the feeling of muscular fatigue.
That is the burn. And if the pH drops too low, the enzymes that power muscle contraction, like myosin ATPase, They just stop working effectively. You hit muscular failure.
So where does carnosine come in to save the day?
The histidine part of the carnosine molecule has a specific chemical structure called an imidazole ring. And that ring is a fantastic chemical buffer.
Buffer.
It can soak up those extra acidifying hydrogen ions, it takes them out of circulation, and helps keep the pH inside the muscle cell stable for longer.
So it's like an internal fire extinguisher for that acid burn.
That's a great way to put it. Having high levels of carnosine in your muscle allows you to work harder and for longer before you succumb to that acidosis. It extends your capacity for high-intensity work.
The text also mentions they activate myosin ATPase and chelate copper.
Right, so not only do they buffer the acid, but they also seem to directly stimulate the enzyme that powers the contraction, and they help manage the levels of metal ions, like copper, within the cell.
There are a couple of clinical notes here, too. One says that urinary levels of 3-methylhistidine are low in patients with Wilson disease.
Wilson disease is a genetic disorder of copper metabolism, where copper builds up to toxic levels. Since these histidine compounds can bind, or chelate, copper, it makes sense that their metabolism would be altered.
And then there's a really scary-sounding one, homocarnosinosis.
A very rare but devastating genetic disorder. It's a deficiency in the enzyme serum carnosinase.
Which is the enzyme that breaks down these compounds.
Exactly. So if you can't break them down, a related compound called homocarnosine builds up, particularly in the brain.
And the result of that buildup.
The text mentions progressive spastic paraplegia and mental retardation. It's a stark reminder that it's not just about making these molecules, you have to be able to clear them out too. The balance is everything.
Okay, let's circle back to something we put a pin in earlier, the idea of the universal donor. We're moving to section 4.
Phenidine.
The methylation powerhouse.
And the star of this section is a molecule called SAM.
S-adenosylmethionine, SAM for short.
Figure 30-7 shows how we make it. It looks like the amino acid methionine gets smashed together with an ATP molecule.
That is a very good, if slightly violent, description of what happens. The enzyme is methionine adenosyltransferase, or MET.
And it uses ATP, the main energy currency of the cell.
It does, and it uses it in a very unusual way. It doesn't just clip off one phosphate group, like most reactions. It clips off all three, splitting ATP into P pi I and pi to drive the formation of this new, very high-energy compound, SAM.
So what makes SAM so special? Why spend an entire ATP molecule to make it? Because SAM is holding onto a methyl group, that's a CH3 group, and it's holding it very, very loosely. That bond is high energy. It's unstable.
And wants to get rid of it.
It is dying to give that methyl group away to another molecule. And that makes SAM the principal source of methyl groups in the body.
So it's the universal methylation agent.
Correct. And this process, methylation, is one of the most fundamental ways the body modifies things. We just saw it to make anserine from carnosine.
And you mentioned it was involved in making the polyamines, spermine, and spermidine.
Yes, the propylamino group actually comes from a decarboxylated version of SAM. But more directly, we use SAM's methyl group to turn norepinephrine into epinephrine, or adrenaline.
Okay.
We use it to make creatine.
Right.
We even use it to modify our DNA in a process called epigenetics, turning genes on and off. SAM is involved in hundreds and hundreds of critical reactions.
It's like the body's universal tool for tweaking molecules to change their function.
It is. It's the ultimate customization tool.
The chapter mentions there are different versions of the MAT enzyme that makes SAM.
Yes, isozymes. Different forms of the same enzyme. MAT1 and MAT3 are found mainly in the liver, while MAT2 is found in most other non-hepatic tissues.
What does that tell us?
It highlights how central the liver is to this whole process. The liver is the main methylation factory, so it has two specialized high-capacity versions of the enzyme to make sure the rest of the body has a steady supply of SAM.
And what happens if the liver is damaged or if these enzymes are defective?
The text mentions you can get hypermethionemia. Methionine from your diet starts to build up in the blood because it can't be converted into SAM. This is a classic sign of severe hepatic dysfunction.
It's just amazing to me that moving a tiny little CH3 group 1 carbon and 3 hydrogens is the basis for so much biological complexity.
It completely changes the shape, the charge, and the function of molecules. It's literally the difference between a neurotransmitter that just regulates blood pressure and the one that initiates the entire fight or flight response.
Well, speaking of neurotransmitters, let's dive into the mood and sleep factory, section 5, tryptophan.
Ah, tryptophan. Everyone immediately thinks of Thanksgiving turkey and feeling sleepy.
So is that a real thing or just a myth?
It's little of both. Biochemically, the link is very real. Tryptophan is the sole precursor for both serotonin and melatonin.
So the link to mood and sleep is definitely there.
Absolutely. The myth part is more about the dosage. The amount of tryptophan in a normal serving of turkey probably isn't enough to knock you out on its own, especially with all the other amino acids competing for transport into the brain. It's more likely the massive carbohydrate load from everything else you ate.
Right. Okay, let's look at the pathway in figure 3010. This is the synthesis of serotonin.
Right. It's a two-step process. You start with tryptophan. Step 1 is hydroxylation via the enzyme tryptophan hydroxylase.
Hydroxylation, so adding an OH group.
Correct. This adds a hydroxyl group onto the ring structure of the tryptophan, creating a new molecule, 5-hydroxytryptophan.
5-HTP. I've seen that sold as a supplement.
You have. And the reason people take it is because that first step, the hydroxylase enzyme, is the slow, rate-limiting step in the whole process. It's the bottleneck.
So by taking 5-HTP, you're basically skipping the bottleneck.
You're bypassing that regulated step because the next step is very fast and simple. It's just another decarboxylation.
Snip off the CO2.
Exactly. And that turns 5-HTP into 5-hydroxytryptamine.
Which is serotonin.
Which is serotonin. Notice the pattern again. Hydroxylate, then decarboxylate. We see this rhythm over and over in making these biogenic amines.
So what does serotonin do? We tend to think of it as the happy chemical in the brain. But the text calls it a potent vasoconstrictor and stimulator of smooth muscle contraction.
That's because while it's famous for its role in mood in the brain, something like 90% of the body's serotonin is actually found outside the brain.
Really? Where?
In your gut, where it regulates motility, and in your blood platelets, where it helps with clotting by constricting blood vessels.
Now, the breakdown, the catabolism is really important here because the text brings up its interaction with drugs.
Right. Serotonin doesn't stick around forever. It's broken down by an enzyme called monoamine oxidase, or MAO.
And what does MAO do to it?
It converts serotonin into a waste product called 5-hydroxyindole-3 acetate, which we then excrete in our urine.
So if you take a drug that is an MAO inhibitor,
You block the enzyme, you stop the breakdown. That means any serotonin that's released stays in the synapse, the gap between neurons, for much longer.
you're prolonging its action.
Precisely. And this was the mechanism of the very first class of antidepressant drugs. They just stopped the cleanup crew, so the room stayed full of serotonin for longer, leading to what the text calls psychic stimulation.
That sounds very 1960s.
It does, but too much of a good thing can be very bad. The text mentions a condition called carcinoid tumors.
What are those?
They're rare tumors that arise from specific cells in the gut called argentaphenoma cells.
And what's special about them?
They go into overdrive, producing massive, massive amounts of serotonin.
What does that do to a person?
It causes something called carcinoid syndrome. The patient gets episodes of severe flushing, diarrhea, bronchoconstriction, even heart valve damage. And the diagnostic clue is that when you test their urine, you find enormous levels of that breakdown product, 5-hydroxyindole acetate.
And from serotonin, we can then make melatonin?
Yes. This next step happens in a tiny gland deep in the brain called the pineal body.
And how does that work?
Serotonin undergoes two more modifications. First, it's N-acetylated, and then it's O-methylated.
Methylated? Wait a minute, don't tell me.
You know it, Sam is involved again. S-adenosylmethionine donates the methyl group.
That's amazing. So you absolutely need methionine and SAM to turn the daytime neurotransmitter, serotonin, into the nighttime hormone of darkness, melatonin.
It's a beautiful piece of chemical poetry. And that melatonin is then released into the bloodstream to regulate our circadian rhythms, telling every cell in the body what time it is.
Incredible. Okay, let's move from mood and sleep to pure, raw adrenaline. Section 6, tyrosine.
The catecholamines.
Yes, and figure 3011 lays out what has to be one of the most famous pathways in all of biochemistry.
The conversion of tyrosine into dopamine, norepinephrine, and finally epinephrine. You can think of it as a ladder of increasing physiological intensity.
Right, let's climb the ladder. Walk us through the steps.
Step 1. We start with the amino acid tyrosine. The enzyme tyrosine hydroxylase adds a hydroxyl group to it.
And that turns tyrosine into...
DOPA, dihydroxyphenylalanine.
Wait, dope as in L-dope, the drug for Parkinson's disease.
The very same. In Parkinson's, the brain cells that produce dopamine die off. So patients can't make enough. We give them the precursor, L-dopa, because it can cross the blood-brain barrier and be converted into the dopamine they're missing.
And this first step, the text says, requires a cofactor called BH4 or biopterin.
Yes, another critical helper molecule. Without it, the enzyme is completely inert.
Okay, so that's step one. What's step two?
Step 2 is turning dopa into dopamine. And for that, we use dopa-deoxycarboxylase.
Our old friend, the decarboxylase? Snip off the CO2.
Exactly. And now we have dopamine, a hugely important neurotransmitter in its own right, critical for reward, motivation, and motor control. But in certain parts of the nervous system and in the adrenal gland, we don't stop there.
We keep climbing the ladder.
We keep climbing. The next enzyme is dopamine beta oxidase. It converts dopamine into norepinephrine.
And this one has some interesting requirements, vitamin C and copper.
Yes, vitamin C is an essential cofactor for this enzyme.
That's wild. I never would have connected vitamin C to adrenaline production. I just think of oranges and preventing scurvy.
Well, this is one of the reasons why severe vitamin C deficiency, scurvy, causes profound fatigue and weakness. You literally cannot synthesize norepinephrine effectively. Your stress response system is crippled.
And finally, the top of the ladder, step 4, turning norepinephrine into epinephrine, which is adrenaline.
Right. And for this final crucial step, we need the enzyme phenylethanolamine N-methyltransferase.
That's a mouthful. But I see the key word in there, methyltransferase.
Which means?
We need SAM again, S-adenosylmethionine.
We do. The enzyme uses SAM to donate a methyl group, sticks it onto norepinephrine, and boom, you have epinephrine, adrenaline, the ultimate fight-or-flight hormone.
So let's just recap that. To make adrenaline, your body needs the amino acid tyrosine, it needs the cofactor BH4, it needs vitamin B6 for the decarboxylase, it needs vitamin C and copper for the next step, and it needs methionine to make SAM for the final step.
If you are deficient in any single one of those nutrients, the entire assembly line grinds to a halt.
That really drives home the idea of nutrient density. It's not just about calories or macros. It's about having all these tiny essential parts available.
Absolutely. The text also makes a brief but important note that tyrosine isn't done yet. It's also the precursor for melanin.
The pigment that colors our skin and hair.
And for thyroid hormones. The text says, in melanocytes, different enzymes hydroxylate tyrosine to form dopamine for melanin.
So the exact same starting material, tyrosine, can become a thought in your brain via dopamine, the color of your skin via melanin, or the hormone that sets your entire metabolic rate via thyroxine.
It's the ultimate choose-your-own-adventure molecule. The fate of that tyrosine molecule depends entirely on which cell it enters and which set of enzymes are waiting for it there.
Okay, let's bring it back to the gym for a minute. Section 7. Creatine and creatinine, figure 3012.
Creatine is famous in the fitness world for its effect on performance, but its biosynthesis is a really beautiful example of inter-organ cooperation.
It takes a village to raise a creatine molecule.
It takes a kidney and a liver, to be precise. No single organ can do the whole job alone.
Okay, that's interesting. Walk me through it.
Step 1 happens in the kidney. The kidney takes 2 amino acids, glycine and arginine.
Okay.
An enzyme called glycine amidinotransferase takes a piece from arginine and attaches it to glycine. This creates a new molecule called guanidinoacetate.
And ornithine is kicked out as a byproduct. More recycling.
Always recycling. Now this guanidinoacetate molecule is then released from the kidney into the bloodstream and travels to the liver.
So on to step 2, which happens in the liver.
The liver takes the guanidinoacetate that just arrived and it performs one final modification. It uses, guess what?
I'm going to go with SAM.
It's always SAM. It methylates the guanidinoacetate to form the final product, creatine.
So creatine is basically a methylated mashup of glycine and arginine?
Structurally, that's exactly what it is. Then, the newly made creatine is released from the liver and travels to the tissues that need it most, primarily muscle.
And what happens in muscle?
In the muscle, an enzyme called creatine kinase attaches a high-energy phosphate group to it, turning it into creatine phosphate.
And this is the energy reserve.
This is the immediate energy reserve. It's like a tiny charged-up battery. When you need to contract a muscle explosively for a jump, a throw, first few seconds of a sprint, you need ATP instantly. You can't wait for glycolysis or oxidative phosphorylation to ramp up.
So creatine phosphate steps in.
It immediately donates its phosphate group back to ADP to regenerate ATP in milliseconds. It is the absolute first line of energy defense for high-power movements.
Now, the text makes a distinction between creatine and creatinine with an IN at the end.
Yes, this is a waste product. Creatine phosphate is not perfectly stable. It spontaneously, without any enzyme, cyclizes and breaks down to form creatinine.
And this happens at a constant rate.
A very constant rate, directly proportional to your total muscle mass. The more muscle you have, the more creatinine phosphate you have, the more creatinine you produce every day.
And we just pee it out.
We do. And because that rate of production is so constant for a given individual, Doctors use it as a clinical marker. They measure the creatinine levels in your blood and urine to assess your kidney function.
Right. If your kidneys are failing, they can't filter out the creatinine, so it starts to build up in the blood.
Exactly. It's the gold standard for estimating what's called the glomerular filtration rate, or GFR. It's basically the health meter for your kidneys.
Okay, on to our final section. We've been ramping up with adrenaline and creatine. Now we need to calm things down. Section 8, GABA.
Gamma-aminobutyrate, the brain's primary brake pedal.
Figure 3013. How do we make the brakes?
It's a masterpiece of biochemical elegance. You start with L-glutamate.
Which is an excitatory neurotransmitter, right? It's the main gas pedal.
It is the most abundant excitatory neurotransmitter in the entire central nervous system, the brain's primary on switch.
And what do we do to it?
You take glutamate and you use one enzyme, L-glutamate decarboxylase. You snip off one CO2 group.
And just by doing that, the gas pedal glutamate becomes the brake pedal GABA.
An instantaneous inversion of function, just by removing one simple carboxyl group. It's a beautiful piece of design.
The breakdown is interesting, too. It's called the GABA shunt. What's a shunt?
A shunt is basically a detour or a bypass. Instead of just being thrown away, GABA could be funneled back into energy metabolism.
How?
GABA is converted to a molecule called succinate semialdehyde. Then, an enzyme called succinate semialdehyde dehydrogenase turns that into succinate.
And succinate is an intermediate in the citric acid cycle, the Krebs cycle.
Exactly. So the brain has this clever pathway where it can take its main inhibitory neurotransmitter, break it down, and feed it directly into the furnace to generate more ATP. It can literally burn its own brakes for fuel if it needs to.
But, of course, there's a metabolic disorder associated with this. 4-hydroxybutyric aciduria.
Yes. If you have a genetic defect in that dehydrogenase enzyme, the succinate semialdehyde can't be converted to succinate.
So it piles up?
Piles up, and the body shuts it down an alternative pathway, converting it into 4-hydroxybutyrate, which is also known as GHB.
Then that's bad.
The accumulation of these metabolites causes a range of mild to severe neurologic symptoms, seizures, developmental delays. It just underscores, again, how critical the cleanup phase is. You have to be able to empty the trash can.
And to finish up, there's a quick mention of beta-alanine.
We saw it earlier as part of carnosine in the muscle. The text just notes it's also formed during the breakdown of pyrimidines, like uracil from RNA, and it can be catalyzed all the way down to acetyl-CoA to be used for energy.
More recycling. Nothing is wasted.
Nothing is ever wasted.
Wow. Okay. We have covered an enormous amount of ground.
We have indeed.
Let's try to synthesize this a bit. We started with this really simple idea of amino acids as just, building blocks. But after going through chapter 30, that feels like calling a symphony just some notes.
It's the complexity in the integration that is so stunning. Think about how a molecule like ornithine is a waste product from making nitric oxide, but it's the starting material for making polyamines.
And how methionine, through SAM, is absolutely required to make adrenaline and creatine and melatonin. These completely unrelated functions are all dependent on this one pathway.
It's a web. It's not a collection of linear roads. It's a deeply interconnected web. And if you pull on one string, say the vitamin B6 deficiency, the whole web starts to vibrate.
Right. Suddenly you can't properly make histamine or GABA or dopamine.
Exactly. The body is an incredibly efficient alchemist. It doesn't invent a brand new tool for every single job. It uses the decarboxylation tool over and over. It uses the methylation tool, SAM, over and over.
And a single starting material, a single amino acid like tyrosine, is responsible for the color of your skin, the control of your movement through dopamine, and your entire survival response through adrenaline.
It really makes you respect the raw materials you put into your body. Every meal you eat is a resupply mission for these microscopic chemical factories.
Okay, so here's my final provocative thought for you, the listener, to mull over. We've talked about all these specific enzymes, the hydroxylases, the decarboxylases, the methyltransferases, and we know from modern genetics that people have slight variations, polymorphisms, in the genes that code for these enzymes.
Right. Some people's enzymes work a little faster, some a little slower.
So could it be that a significant part of my personality, maybe I'm a naturally more anxious person, or I'm a really deep sleeper, or I recover from exercise faster than my friend, is actually just a direct reflection of the microscopic tuning of these very enzyme factories?
Oh, that's almost certainly true to a large extent. We attribute so much to high-level concepts like willpower or character. But at the end of the day, if your version of phenylethylamine N-methyltransferase is a little bit sluggish.
The enzyme that makes adrenaline?
Yes. You might literally be a calmer person because your body can't spike its adrenaline levels as high or as quickly as someone else's. Or if your tryptophan hydroxylase is natural, very efficient, maybe you maintained higher serotonin levels and are just, constitutionally more optimistic.
We are, in a very real sense, just walking, talking chemical reactions.
We are, but understanding how those reactions work, that gives us a little piece of the owner's manual.
And that is a very powerful thing to have. Thank you for taking this deep dive with us.
My pleasure.
Go eat some protein and just take a second to think about the incredible alchemy that's happening inside you right now. Until next time on The Deep Dive.